Okay, this video is going over some major differences between B cells, T cells, humoral immunity, and cell-mediated immunity. So to first look at the differences between innate immunity and adaptive immunity. Innate immunity would be things like your skin, so your epithelial barriers aren't letting these allergens or microbes, antigens, whatever you want to call them, in past the skin. So that is a first barrier. Phagocytosis is when your cells eat the microbes or the antigens, and then your non-killer cell or your natural killer cells, which I will go into in a little bit. Those are all, and the complement cascade. Those are all innate immunity functions. Adaptive immunity functions is the humoral immunity and the adaptive immunity, and the cell-mediated immunity. Humoral immunity is your B cells. So always remember, humoral is your B cells. And I remember that because humoral kind of sounds like human. So human beings, we are human beings. Being starts with a B. Humoral beings. Humoral immunity is a B cell activation. Cell-mediated immunity uses T cells. So cell-mediated immunity uses T cells. So that is a big difference between humoral immunity and cell-mediated immunity. B cells, I think I have them over here. So both B cells and T cells are made, they're different white blood cells. They both are. B cells, their main function is to recognize antigens. So B cells will eat anything that is considered non-self. So if your B cells recognize this green dot as an antigen, so it says, hey, that's not something that belongs to me. That's not part of me. I'm going to eat it. This is a nucleus in the B cell. The B cell will then eat the antigen and break it up. The broken up pieces will then be distributed, and this is a MHC2 cell, protein that will show. So say you broke down this antigen into tiny bits. You took one little bit that you broke off, and it's going to show it on the outside of the cell on this MHC2 receptor. The MHC2 receptor will bind to a CD4 cell or a helper cell. And then it goes through I'll go through this in a little bit more detail, but then the CD4 cells release lymphokines that want to produce more B cells to go through this process, more and more and more. And then the B cells are converted into plasma cells, which will give you the antibody to this specific antigen that you put out, saying that this is foreign. Let's make an antibody to this. And that is what happens in the end. The B cells get converted into plasma cells, which kind of spit off antibodies that the B cells were showing on their MHC2 receptors. Now, T cells can be differentiated into a few different types of T cells. I think he goes over CD4 cells and CD8 cells. So CD4 cells are helper cells, and those bind only to the MHC2 receptors on any kind of cell that is a antigen presenting cell. An antigen presenting cell is anything that presents antigens. So like these B cells did right here, it's presenting an antigen. That is an APC, antigen presenting cell. So macrophages, B cells, and another one, dendritic cells. They are all able to eat a antigen and then present it via the MHC2 receptor. So back to T cells. T cells, the CD4 will combine to the, I labeled these up here. CD4 will only combine to the MHC2 receptor. CD8 will combine to MHC1. And you can remember that, which one goes with which, by using a rule of 8. So always have the number 8 in mind. So if your CD8 only combines with your MHC1, if you multiply 8 times 1, you get 8. And if your CD4 only combines with your MHC2, 4 times 2 is 8. So that's a quick, easy way to remember which T cell goes with which receptor here. So CD8 cells, so this, let's start at the beginning. So you have a healthy cell, you have an antigen. Your healthy cell, say that's any cell that will eat it. So a dendritic cell or a macrophage, so something that's going to eat it. That cell has eaten the antigen, it's breaking it down. And then every single cell that has a nucleus has an MH1 receptor. Every single cell has an MHC1 receptor, every single one. Only cells that are able to eat other cells like macrophages, dendritic cells, and B cells will have the MHC2 receptor. So split that up in your mind. Every single cell will have the MHC1. Only cells that are able to eat other cells will have MHC2. So here I put both out there because this is a cell that is, A has a nucleus and B can eat another cell. So that's why it has both. So our cell ate the antigen, broke it up, spit out pieces of that antigen, that invading body, onto the MHC1 and MHC2. So then CD4, the helper protein, came along and attaches to this MHC2. What the helper, CD4 is called the helper. CD4 will break off and either release TH1 or TH2. TH1 will release interleukin 4, which stimulates the B cell activation, which the B cell activation was what I just went through, where the B cells will take the antigen, break it up, show the MHC2, and then break it down, produce antibodies. So that is that pathway. But another quick math one. If you take the whatever interleukin number, see how TH1 releases interleukin 2 and TH2 releases interleukin 4. Divide the interleukin number in half. So interleukin 4 gets released by TH2, interleukin 2 gets released by TH1. So say you come down this way, interleukin 2 is released, that will stimulate CD8 cells. CD8 cells are cytotoxic cells. So just the name in itself it's cytotoxic to other cells. So CD8 will recognize, it'll be on the lookout for cells that have the MHC1 of this foreign antigen piece on the outside. Because remember, it's just a piece that's on the outside. It'll recognize, say, hey, that's not what's supposed to be here. And then the CD8 breaks the plasma membrane and then the cell dies. So the T cells go through this whole process.